Now I am explaining gametogenesis. Gametogenesis is the production of gamete from sexual reproduction. This process takes place in germ cells present in the primary sex organs, the testes in the male and the ovaries in the females. Gametogenesis is controlled by gonadotropic hormone of anterior pituitary. Spermatogenesis is the production of sperm in the testes and oogenesis is the production of ova in the ovary. They are commonly called gametogenesis. I am repeating. Gametogenesis is the process of gamete formation in sexually reproducing animals. Gametes are produced in the gonads by a series of changes in their germinal epithelial cell. The formation of male gamete means spermatozoa in the testes is called spermatogenesis, and the development of female gamete means ova in the ovaries is said to be oogenesis. Now I am talking about spermatogenesis. Spermatozoa are produced in the seminiferous tubule of the testes. This process includes two stages. Number 1, formation of spermatid. Number 2, metamorphosis of spermatid. Number 1, formation of spermatid. Spermatid are formed by three phases, namely phase of multiplication, that is mitosis, phase of growth, and number 3, phase of maturation, means meiosis. During phase of multiplication, the primordial germ cell or PGCS divide repeatedly by mitosis to form diploid spermatogonia. Each spermatogonium is diploid and contains 46 chromosomes, divide mitotically into two primary spermatocytes. During phase of growth, the primary spermatocyte enlarges in size and prepare to undergo meiotic or maturation division. During phase of maturation, the primary spermatocyte undergo meiosis 1, giving rise to two haploid n equal 23 secondary spermatocyte. These cells remain connected by cross bridge of cytoplasm after each division. They further undergo meiosis 2, meiosis 2, resulting in the formation of 4 haploid spermatid. I am repeating. Spermatocytogenesis. The cell of germinal epithelium of seminiferous tubule of the testes which enter the process of spermatogenesis are called primary germ cells. These cells are basically similar to any other body cell in having a diploid number of chromosomes. Characteristics of the species. Each primary germ cell enters a multiplication phase and by repeated mitotic division give rise to a number of cells. These unspecialized male cells called spermatogonia also continue to divide for some time and then enter the growth phase. In this phase, spermatogonia grow and increase in size. During spermatocytogenesis, each spermatogonium divides mitotically to form primary spermatocytes, which are diploid and interconnected by their cytoplasm. Meiosis 1. The primary spermatocyte with large spherical nuclei undergo the first meiotic or maturation division, each forming two haploid X secondary spermatocytes. Meiosis 2. Each secondary spermatocyte undergo the second meiotic or second maturation division and produces two spermatid. Thus, each spermatogonium gives rise to four interconnected spermatids. A spermatid is a rounded cell with a spherical nucleus. Next one is spermiogenesis. The process of transformation of a circular spermatid to a spermatozoa is called spermiogenesis. I am repeating. The process of transformation of a circular spermatid to a spermatozoa is called spermiogenesis. In this process, the nucleus of spermatid becomes the head of the spermatozoa. The Golgi apparatus of the spermatid containing proteolytic enzyme becomes the acrosomal cap. Spermatozoa are situated most centrally in the seminiferous tubules. Spermatogonia most peripherally, and others such as spermatocyte and spermatid occupy intermediate position. Next, spermiation. After spermiogenesis, sperm had become embedded in the Sertoli cell and are finally released from the seminiferous tubule by the process called spermiation. In men, the entire process of spermatogenesis takes about 74 days. It starts at the age of puberty due to significant increase in the secretion of gonadotropin releasing hormone GnRH and continues even in the old age. Now I am explaining metamorphosis of spermatid. Spermiogenesis is the term used to describe the conversion of spermatid to spermatozoa. This process takes place within the cytoplasm of Sertoli cell and does not involve any cell division. A spermatid is non-volatile and heavy with organelle like mitochondria, Golgi body, centriole, nucleus, etc. During spermiogenesis, the weight of gamete is reduced along with development of structure for motility and penetration. After spermiogenesis, sperm leads become embedded in the Sertoli cell and are finally released from the seminiferous tubule by the process called spermiation. Spermatogenesis starts at the age of puberty due to significant increase in the secretion of gonadotropin releasing hormone GnRH from hypothalamus. The increased level of GnRH then acts at the anterior pituitary gland and stimulate secretion of 2-gonadotropin, luteinizing hormone LH, also called interstitial cell stimulating hormone ICSH, and follicular stimulating hormone FSH. ICSH acts as interstitial cell of Leydig and stimulates synthesis and secretion of testosterone. Testosterone in turn stimulates the process of spermiogenesis. FSH acts on the Sertoli cell and stimulates secretion of some factors which help in the process of spermiogenesis. A typical human sperm is flagellated, consisting of 4 parts, namely head, neck, middle piece and tail. The nucleus becomes compact, forming the major part of the head, spermatozoa. Golgi complex of spermatid give rise to acrosome, which form a cap in the front of nucleus containing a lytic agent. Acrosome of mammalian sperm produces sperm lysine called hyaluronidase, which dissolve egg membrane. If acrosome is removed from a sperm, it will fail to penetrate into the ovum. There are 2 centrioles in the spermatid which become arranged one after the other behind the nucleus. The anterior one, the proximal centriole, is usually located on the neck of the spermatozoa. During fertilization, it is introduced to the egg and is required for the first cleavage. The posterior one, the distal centriole, changes into basal body, giving rise to the axial filament of the sperm. The middle piece possesses numerous mitochondria. They provide energy and strength for the movement of tail that facilitate sperm motility, essential for fertilization. तो इस तरीके से हमने देखा कि जो स्टेप हैं स्पर्मेटोजेनेसिस को, उनको हम डायग्राम से रिप्रेजेंट कर सकते हैं. मल्टीप्लिकेशन फेज, फिर ग्रोथ फेज और फिर मैचुरेशन फेज में. तो पहले प्राइमरी जर्म सेल जो होती है उसमें डिप्लॉयड क्रोमोसोम होते हैं जिन्हें 2x से रिप्रेजेंट किया जा सकता है. फिर ये स्पर्मेटोगोनिया जो है इसमें कन्वर्ट होकर पहले प्राइमरी स्पर्मेटोसाइट यानी कि 2x में फर्स्ट मैचुरेशन डिवीज़न होता है और सेकेंडरी स्पर्मेटोसाइट बनते हैं जो कि हैप्लॉयड इन नेचर यानी कि x होते हैं. फिर इसमें सेकेंड मैचुरेशन डिवीज़न होता है तो स्पर्मेटिड बनता है और स्पर्मियोजेनेसिस की प्रोसेस से स्पर्म का फॉर्मेशन होता है. Now I am talking about factors affecting spermatogenesis. A number of factors influence spermatogenesis. The gonadotropin releasing hormone of hypothalamus, that is GnRH, stimulate the anterior pituitary, which then stimulate the 2-gonadotropin hormone, number 1 luteinizing hormone LH, and follicular stimulating hormone FSH. The LH, also called ICSH, act at the Leydig cells and stimulate the synthesis and the secretion of testosterone androgen from the testes. Testosterone also stimulates spermatogenesis. FSH act on the Sertoli cell and stimulate the secretion of some factors which help in the process of spermiogenesis. However, high temperature is injurious to spermatogenesis. The fact that testes hang in the scrotal cavity where the temperature is comparatively low than the abdomen is thus technologically desirable. The testes that lies within the abdomen due to failure to descend means undescended testes fails to manufacture spermatozoa. A fully formed spermatozoa pass through the male genital passage. They undergo a process of maturation. They acquire some motility only after passing through epididymis. The secretion of epididymis, seminal vesicle and prostate have a stimulating effect on sperm motility, but they become fully motile only after ejaculation.